dystrophies. So I would now invite Dr. Sandona, a researcher at the University of Padova at the biology department, to introduce her study that was financed by Teleton last year as regards misfolded proteins, a new approach for the treatment of sarcoglycanopathies. Well, I hope you can see me. Well, first of all, I want to really heartfully thank Mrs. Vola and all the patients and the relatives of the different patients who so strongly wanted this meeting, because I believe that putting together researchers working on rare diseases like sarcoglycanopathies is very important to network for us to get within short to identify a treatment for these diseases. I also want to thank the people who work with me, particularly Lisa, Roberta, Chiara, Ilaria, and those who physically financed our research at Teleton, IFM, and the University of Padua. I put on this slide because you all know what the story is, but what I wanted to show you here is simply the situation that we can view when analyzing the muscle in a healthy person. You all know that all sarcoglycans are all correctly localized at membrane level on the skeletal muscle, associated with all the right proteins connected to dystrophin. But what happens when there is a mutation in the genes codifying the sarcoglycans? In this case, the example is the alpha sarcoglycan codifying gene, and in this case the beta sarcoglycan. We are in front of the loss of the mutated protein, the alpha sarcoglycan, or at least a strong reduction in the mutated protein. What happens is that the rest of the complex disappears as well, or is strongly reduced. So what I want to say with this is that a fault in one protein actually strongly reflects also on the other sarcoglycans. And the other question is that evidently the sarcoglycan complex works as a structural functional unity within the muscle. What are the mutations that hit sarcoglycans? Lots of types. Some of them impede the production of the protein. In other cases, we have shorter proteins than usual, and this is the example of the beta sarcoglycan, the most frequent example in Italy hitting beta sarcoglycan. In many more cases, however, the gene mutation brings to a very small change in the protein. The protein has normal size, but it presents a single amino acid substitutions. And this quite often may lead to impacts on the structure of the protein, but not that much on its function. In some cases, it might be functional yet. The problem, however, is that the mutated protein and all the others disappear or they strongly reduce. Another thing to remember is that the level of messenger RNA, that is the molecule that from the DNA, the gene, mediates to then reach the protein, that is at almost normal levels in people affected by sarcoglycanopathies. So the problem that leads to the loss of our mutated sarcoglycan is something that occurs downstream. What might that be? Well, you all know that all the cells, and the muscle is no exception, contain a quality control system that is very sophisticated and that checks all the new proteins that are synthesized, and only if the structure of these proteins is correct, these are sent to perform their own function. However, if this structure is not reached within reasonable times, the system decides that they are to be eliminated to avoid the accumulation of possibly toxic, damaging proteins. And the system checking the control quality are the ubiquitin proteasome system, which is added a sort of flag to the protein that must be eliminated, and that has then the degradation system recognize it. That is the proteasome. The other strategy used by the cell is that of autophagy, but Dr. Fimia will speak immediately after me exactly on this. However, through these two systems that are strongly correlated, interrelated, misfolded proteins are eliminated, and with them also the proteins that have a normal structure but which cannot find their own partners, for instance wild type sarcoglycans that disappear together with a mutated sarcoglycan. A few years ago, both us and Dr. Richard, as you heard before, demonstrated that sarcoglycans, mutated sarcoglycans, are substrates of the ubiquitin proteasome system. So, the key event in the sarcoglycanopathy disease is a premature degradation of the mutated sarcoglycan. And even though the sarcoglycan might be still functional, this is eliminated, thus bringing to a de facto loss of function. So the question is it possible to restore a healthy phenotype? Well, today we heard lots of speakers telling about interventions that aim at giving a healthy copy of the gene to the patient through very innovative therapies like gene therapy or stem cell therapy. Our approach is different. What we would like to do is try to cure what is already within the cell, that is, the mutated sarcoglycan. And we have the idea to follow this path through two strategies. On one side, we would like to restore the protein as Dr. Richard already does by trying to reduce the mutated protein degradation. On the other side, the second strategy we would like to follow actually regards the use of molecules that might help the protein which is not able to correctly fold to find a structure that mostly resembles that of a normal protein, thus promoting the own correct localization. Right, to understand where and how to act, I'll show you this slide that Dr. Richard already showed to you. But this is only to say that when sarcoglycans are produced, and they are normal, so to say, they are helped at ER level to fold correctly. Glucids are added, and many more structures are created. They assemble the whole complex and they are localized at membrane level. However, if the folding does not take place in correct time and in due form because of a mutation, the control system actually tries to help with further folding cycles on the protein, but if they do not produce any results, the system controls that the protein is to be degraded and thus sends it to a whole series of proteins intervening in what is called the degradation of proteins associated with the ER, and this brings proteins to be brought back within the cytoplasm, where, however, they are flagged by ubiquitin and then they are degraded by the proteasome. Our first strategy would like to block this path, right, to be able to move the balance of the pathway towards the possibility of reaching at least a partially normal folding, thus bringing the protein where it has to be on the cell's membrane. A few years ago, we published a study in which, by blocking the final step, that is the proteasome, we were able to obtain this result. And this is simply to show you that we have tested this on several mutants of alpha sarcoglycan, and the protein, by blocking the proteasome, accumulates, and not only does that accumulate, but it also ends up to be where it has to be localized at cell membrane level. Of course, these are studies on heterologous cells. These are not muscle cells, but very similar results were also obtained on a small fragment of a biopsy from a patient hit by LGMD with these two mutations on the two alleles. And again, in this case, a treatment with an inhibitor, which is used in clinics to treat multiple myeloma, brings to an absence of the expression of the protein, to the recovery of the expression of the sarcoglycan, alpha, beta as well on the surface. So this lets us hope for the positive result that our approach may not only work on a cellular in vitro system, but also on the physiological side of the muscle. However, we well know that blocking the proteasome may have some collateral effects that are so considerable that they might bring not to our treatment not to be effective. So we started a whole new work in which we are trying to identify, well, we already have had some positive results, so identifying all the components that intervene before the protein leaves the reticulum, thus without blocking the final step, but through blocking the intermediate steps, so that we can act on more specific sites, thus reducing possible severe side effects that might instead occur through a systemic block. To this purpose, of course, the identification of these elements make them pharmacological targets, and Sikanower, the Nobel Prize winner, says that there are at least 1,500 different factors that might intervene in the degradation of proteins connected to the ubiquitin proteasome system. And there are at least 800 different ligases proteins that may be specific targets. So we have a very high potentiality in this sense. To cut it short, we have used the first approach to see who acts in the degradation of the mutated sarcoglycan using RNA interference to reduce the expression of these ERAD components. And as you can see here, we have already identified a series of them. These are proteins that actually help folding the proteins. This is a protein that acts as an intermediary, taking the mutated protein and bringing it to the proteins that bind the ubiquitin. By blocking, reducing the expression of these proteins, we increase the amount of mutated alpha-sarcoglycan. We have also adopted other methods, so we introduced in cells expressing the mutated alpha-sarcoglycan, in this case this mutation here, some mutants, dominant and negatives, of ERAD components. Here we have acted on this ATPases, which is a protein providing energy to extract from the proteins from the reticulum, thus thereafter sending them to the proteasome. And if we use these negative dominants that are able to block the function of the endogenous therapy, we are able to recover mutated alpha-sarcoglycan. In the end, we did that actually on several proteins on the reticulum. However, one more approach we used to see whether physically these proteins interact, as I showed before, with the mutated alpha-sarcoglycan. We actually produce an evidence of immunoprecipitation. So we use an antibody that is able to capture the alpha-sarcoglycan, and together with this protein, we can actually recover other ERAD elements that are fundamental for its degradation. So, to get back to our scheme, if we act on these elements, well, this might be a crucial point. And I won't show you the experiments because we are carrying them out during these days, but we have recovered from some Japanese studies some specific inhibitors, E3 ligases that I showed before, for them to interact, and the experiments we have underway are very promising in terms of increase in the protein and localization on the right side on the membrane. Very two last things I want to tell you about. This new strategy, again, that actually wants to act in a fully different way. In this case, I'm not blocking a component, but I'm helping the folding of the protein. And to help the protein fold, I use small molecules, chemical molecules, that can actually interact and bind with the folding protein, thus helping the same protein and promoting it on this path. We did this by using some molecules that are already well known. As you see here, I gave them simple names A, B, C, and so on and so forth, because in this moment I cannot tell you the name of the molecules, because we are trying, they are being patented, their use is being patented. What I only want to show you in here is that in this case, the alpha-sarcoglycan mutation is this one here. If we treat the cells expressing this mutation through these substances, you see that in most cases, and in these two for instance, we can actually obtain a level of expression which is comparable to that of the wild type. This would be the mutant level, this would be the wild type level. An interesting thing to say is that depending on the concerned mutation, the best result is obtained through different molecules to show that the site where the mutation is important for these molecules to act. And in the end, very last thing I wanted to show you was that these small molecules do not only help accumulate the mutated protein, but the accumulated protein also accumulates on the right place, so on the surface. Please note that this is one of those alpha-sarcoglycan mutations that, for instance, in the pharmacological strategy as proposed by Dr. Richard, was not functionally recovered on the membrane. We are currently producing the functional measurements, measuring the adipiasic functionality and values of the protein that are extremely promising. Very last slide, just to say that we strongly believe in these two approaches, and the first data we have gathered are very promising. Of course, we have a whole lot of work to be done yet. First, we want to test these approaches in a situation which is not only a hack cell expressing the sarcoglycans, for instance, myotubes, particularly from patients affected by sarcoglycanopathy. And to this purpose, I really want to thank Professor Comi and Professor Angelini, whom we are working with. Of course, we are trying either to produce or to obtain mice that might act as models to study these approaches in vivo. And here there are a few problems to be solved, actually, as also Dr. Richard told you, because it is not easy to have that model. Knockout mice have no protein. We need mice that have the protein with the mutation to be able to check that our system actually works. Of course, we are looking for and testing and screening new molecules, right, to help either inhibit the Arab components or help the proteins folding. And of course, one more fundamental aspect, this approach is potentially applicable not only to alpha-sarcoglycan, as I said, but to all sarcoglycans presenting a mutation bringing to a loss of the protein, but not of its functionality. And to this purpose, we have created different missense mutants with a single replacement of the beta sarcoglycan, and the first experiments seem to go the same way. However, we are also working on very different proteins, for instance, the adipiasis and the sarcoplasmatic reticulum, SERCA, or cardiac cell calsequestrin. Thank you for your attention.
Domande?
Is there any question?
No, più che una domanda è un'osservazione.
This is an observation. The knocking, as far as the alpha-sarcoglycan is concerned, cannot work, may not work, because the conservation of the alpha-sarcoglycan is very low, and there is also the problem related to the exon and so on. So the rise genes where we can work on are the alpha and beta sarcoglycan. So the mutations should be those in order to verify if your experiments are right. Yes, of course.
Because as a clinician, I think it's a very attractive therapy, the chaperones therapy. And we have the example of Pompe disease, which is another, but it's a metabolic disease where the chaperones are currently tried. And I think that it could be very interesting, but we have to be also careful because there was a first trial of the chaperones alone with a worsening of the disease. 2 patients were included in a big protocol, and these 2 patients worsened, but probably because there was a question of dose, of excessive dose, and due to the link with the enzyme, it was not an improvement of the enzyme activity, but probably a decrease. But now there is another trial combining chaperones and enzyme therapy, which could be very promising. And I think for this disease, it's also very interesting.
Yeah, I just want to make a comment about the rescue of sarcoglycan that Sandona showed so beautifully, you know. And this is certainly possible when there is misfolded proteins and there is an inhibition of the degradation, and eventually they get rescued to the sarcoplasma, like she beautifully showed. The drug that has been used, that is Velcade, has been used in multiple myeloma, and therefore we could theoretically use it. The problem is that the dose that is used in multiple myeloma, I mean, is toxic for the patients. So although the substance is theoretically at disposition, and we could order it tomorrow, you know, and it works in vitro in certain type of mutation, I mean, to put up a clinical trials, you know, is a question of safety for the patient, you know. So we need a better drug that inhibits this ubiquitin proteasome and brings the sarcoglycans to the surface, because once they are to the surface, I know that Doriana is working very hard to look at the molecules that show up, the complex that show at the surface. She didn't say, but she's working on the ecto-ATPase function of alpha-sarcoglycan. So eventually, once we make the core, like Vincenzo said, of beta and delta sarcoglycan, and then we add back the alpha, you know, and we put it as sarcoplasm, something should happen also functionally there, you know. So this is a pathway that theoretically could be followed, but it's not the only one.
Is there any other question? We would like to thank Dr. Sandona